ahead and get started here. It's my pleasure to introduce Dr. Lorenz. Dr. Lorenz is an assistant professor in the Department of Agronomy and Horticulture at the University of Nebraska-Lincoln. He received his bachelor's degree in plant science from the University of Minnesota, his master's degree in plant breeding from right here at Iowa State, and his PhD in plant breeding and plant genetics from the University of Wisconsin. He's going to talk to us today about prospects for dissecting drought tolerance in maize using high-throughput phenotyping. So let's welcome Dr. Lorenz.
Thank you, Gerald. And I'm glad to get to start a little bit early here because I have a lot of slides. And it's great to be back here in Ames. I don't get back here nearly enough. And like Gerald said, I'm a proud graduate of this program. So let's just get started. So most of the time when I'm asked to give a presentation, I'm asked to talk about genomic selection. I would say most of my research and most of my time is spent thinking about genomic selection. So I'm going to give you a whirlwind tour of genomic selection, some of the things that we're doing right now. And then I'm going to tell you, I prefer to do genomic selection all the time, but I'm going to tell you a little bit about why I'm interested in high-throughput phenotyping for improving plant breeding. And then I'm going to tell you about some of the things we have going on in this area. And I should mention that I'm not published in this area at all, and so the research really in this area in my program is at a very nascent phase. So this is the trend. We're all very familiar with this, and this is the reason why we're interested in using genomic selection in plant breeding to increase the efficiency and effectiveness of plant breeding. Now, I should point out that if the organizers of this symposium have their way, this would not be the situation, but I'm going to put my money on this situation. So to make plant breeding most cost-effective, we should maximize the successful use of genomic information in those breeding programs while minimizing the amount of phenotypic data we need to collect to use that genomic information. So genomic selection is a pretty popular topic these days, and so I don't have to go into the basics in much detail. But essentially what we have is we have a training population, or sometimes called a calibration data set, where we take the phenotypic data, and we take the genomic data, and we combine those together, and we build some sort of statistical model. Okay? And then we use that statistical model to predict individuals that have been genotyped, but not phenotyped. Okay? And the nice thing about this is that if the genotyping is a lot cheaper than the phenotyping, we should be able to increase the number of individuals flowing through our selection program. We can make genomic predictions during the off-season and thereby increase the genetic gain per unit time, perhaps. And if our genomic predictions are actually more accurate than our phenotypic evaluations, we can increase our selection accuracy. So there are lots of reasons why we might want to use genomic selection in plant breeding. And I'll say the beautiful thing about genomic selection is we don't have to worry about QTL mapping. We're not doing any testing for significant markers. Okay? We're simultaneously using all the information available to us. And since we're using all the information available to us, and we often, in the age of high-throughput genomics, we oftentimes have many, many more markers. And we do observations, we end up what statisticians call the large P small n problem. And so we have to use a series of more sophisticated statistical models to deal with this problem. Just using ordinary least squares for estimating our marker effects is no longer sufficient for lots of technical reasons. I'm not going to go into much detail on this right now. A good reason for using genomic predictions as opposed to a traditional marker-assisted selection approach is that it often works a lot better. So the study by Lorenz and Bernardo back in 2009 looked at about 36 different instances of trait and population combinations, and you can see that every single time genomic predictions worked, in terms of prediction accuracy, worked just as well and oftentimes much better than prediction accuracies obtained from marker-assisted selection. Okay? And then some simulations of my own also show a strong advantage of using genomic predictions for making selections compared to a marker-assisted selection approach. Another reason why plant breeders really like genomic predictions is because they can get the predictions and they look just like their phenotypic data they're used to dealing with. Okay? So they can take these predictions and put them in an Excel format and color, do some conditional formatting and sort them in whichever way they want and make their selections. They don't have to worry about individual A having marker allele 1 and so forth and all different types of combinations. So it's easy to deal with. So this is what the process looks like in full motion. We have what we call the line development cycle over here, where we're creating progenies, we're genotyping progenies, and we're making selections based on those predictions from our statistical model that we're building over here in the model training cycle. Okay? And this consists of phenotyping some set of individuals derived from our line development cycle, okay? Phenotyping them in relevant environments, and updating our prediction model, okay? To use so that our prediction model contains or is made from a calibration data set that contains individuals closely related to our selection candidates as we can. Because as I'll show you, there's a very important relationship between the genetic relationships of individuals in our calibration data set and our selection candidates that we're trying to predict. So surrounding this process, I like to use this slide because surrounding this process I can outline a number of questions that I'm interested in. And I'm interested in helping plant breeders get straightforward answers to these common sense questions. So questions like, what marker platform should I use? Okay, if I should use genotyping by sequencing, is there any way I should filter those markers, okay, before I use them in predictions? Which lines do I advance from my line development cycle to my model training cycle? Okay? Are there lines that are more informative for model building than other lines? And what about experimental design? So as we go from wanting to estimate the allelic effects, or wanting to go from estimate whole genotype effects to estimate allelic effects for genomic predictions, our equation in terms of balancing resources across replications, locations, and individuals completely changes. Okay? And I'm not going to talk much about this today, but I had a paper out about a year ago that looked at this topic in terms of allocating resources between population size and replication. And on top of that, which statistical model should I use? And so I'm interested in working with plant breeders, particularly I work with a lot of public plant breeders in helping to answer some of these questions as they think about embarking on incorporating genomic prediction into their breeding programs. So I want to point out that everything I'm going to present to you in the next 10 or 20 slides doesn't have anything to do with any sort of genomic function, functional genomics, genetic architecture, or anything like that. Okay? So just beware, let alone phenomics. So first question here, what about marker platforms? So we have genotyping by sequencing versus maybe a 92,000 or an Infinium assay that includes 92,000 SNPs, okay, for hard red winter wheat. And to pursue this project here, I'm working with Steve Benziger, the wheat breeder at University of Nebraska, and the genotyping was done with these two different marker platforms by Edward Akinov and Jesse Poland at Kansas State. Okay? And we had the phenotyping is done in Nebraska in 2012-2013. Some good replication and a good standard procedure of cross-fold validation, replicated 100 times was used. So this is the situation for the markers. So the thing about genotyping by sequencing is that it's cheap, but there's a whole lot of missing data that comes along with these markers. Okay? The Infinium assay, this is a mistake here, the Infinium assay gives you very clean data, but it's fairly expensive. So you can see the missing data rates and the minor aloof frequency of these different platforms here in this slide. So the take-home is that in the Infinium assay, we had about 28,000 polymorphic SNPs that had less than 50% missing data, which there are very few missing data points in the Infinium assay. Okay? With the genotyping by sequencing, we had 20,000 SNPs that had a minor aloof frequency greater than 0.05, and like I said, a lot of those SNPs had a whole lot of missing data. And the person working on this project is a postdoc named Ibrahim Al-Bassiani. Okay, so these are the results. So we have, what I want you to see here is basically compare the dark blue bars to the lighter blue bars. Okay? You can see that in all these different situations, in terms of models and trait and so forth, the genotyping by sequencing does just as well as the Infinium assay, okay, if not better. So, and to emphasize that we don't know anything about the trait here for genotyping by sequencing, we wouldn't even know where these markers are. We have no idea where these SNPs are located in the genome. We can know, according to a genetic map, where the SNPs fall for the Infinium assay. But there's no physical map, complete physical map for wheat, so these are just used as cofactors in a model. Okay? But it's useful for prediction. Okay, so what about soybean? Working on George Graff, the soybean breeder at the University of Nebraska. We have about 300 lines in this training population. Traits, granule, plant type, maturity date. Again, genotyping by sequencing has worked fairly well, so we're going to go with this method of genotyping for this project. And we have about 219,000 SNPs that could be called if they were polymorphic in this population. Okay? And again, standard cross-validation procedures were used to study this for this study. This is what the situation looks like in terms of the distribution of SNPs across the soybean genome with genotyping by sequencing. There's not a lot here that's relevant to genomic predictions. You can see that, as we'd expect, the SNP number is higher towards the ends of the chromosomes. Missing data frequency is higher towards the center of the chromosomes because of increased repetitive development. And you can see that some chromosomes have more diversity in terms of minor allele or measured by minor allele frequency than other chromosomes. But like I said, there aren't a lot of implications here for genomic prediction, but I think just it's a thorough job to present this data anyway. Okay, so if we go look at the number of SNPs in this population measured here, or scored by genotyping by sequencing, you can see that the number of SNPs falls off rapidly if we make a cutoff based on amount of missing data. At the end of the day, what we have is about 16,000 SNPs that have less than 5% missing data that are polymorphic in this population. If we include everything, up to 80,000 or up to 80% missing data, SNPs that have 80% missing data, which just sounds absurd, but we can use them. We have 52,000 SNPs. So okay, here we go. So the question here is, we have all these SNPs, we're awash in SNPs. Is there some sort of filtering mechanism we should use to reduce our marker data set size and perhaps get better predictions? Well, what I have here are slides here, or figures here that show the correlation between the predicted and the observed in the field and the minor allele frequency, and then I have a separate line for each of these levels of missing data. Okay? And what you'll see is that the lines are pretty straight. Okay? So if I cut off markers based on minor allele frequency, I don't have a whole lot of effect in terms of prediction accuracy. And the lines are also quite close together. Okay? So basically it says that I can include SNPs that have up to 80% missing data and still get quite good prediction accuracies. Okay? So this indicates that filtering is not much to worry about in terms of filtering. We go a step further and we look at the effect of imputation. So we're missing a lot of this data, but there are fancy methods out there that we can use to predict what those marker scores would have been if we could have observed them. One of those methods is called a random forest imputation. And if you look real carefully here, you'll see that there's a slight increase in prediction accuracy. So if we impute all that missing data, up to 80% missing data, we get a very, very small bump in prediction accuracy. Okay, so once again, we know where these SNPs are because there's a complete physical map for soybean, but we're including lots of missing data here, but we're still getting good prediction accuracies on things like yield. Prediction models is a big question all the time in genomic prediction. I think in plants, it's more or less been shown again and again and again that there's no difference in terms of prediction model. There are really good theoretical reasons out there that I won't go into, but Daniel Gianola had a paper on this in Genetics not too long ago. It has to do with the number of individuals that were evaluated oftentimes in these plant programs. But they're oftentimes very equal in terms of prediction accuracy. So we can go a step further and we can start looking at epistasis. There are lots of different ways to include epistasis in a genomic prediction model. This is just one using some sort of a genome-wide modeling approach. And so basically what we're doing here is we're taking the element by element matrix product of the gene matrix. Okay? So if you think back to introduction to quantitative genetics, you remember that the genetic covariance between two individuals is a function of the additive relationship. Okay? 2 times the coefficient of coancestry squared. And then you look at the component coming from the additive by additive epistasis. That's 2 times the coefficient of coancestry, 2 times the coefficient of coancestry squared. Okay? So you might think that taking the additive relationship matrix here and taking the element by element wise product of those two will give you something for explaining epistasis in this population. Okay? And there's a better way to do this actually. That was presented by Zhu here fairly recently in a genetics paper that we looked into. But skipping over lots of details, I just want to point out that we also saw no difference in terms of prediction accuracy when trying to model epistasis. To make a long story short. Okay, all right, so we have marker platform, marker filtering, a little bit about on prediction model. What about the genetic relatedness between the training population and a set of selection candidates? Okay, so for this project, I like to use this data set collected in collaboration with, or collected by Kevin Smith, and I'm a collaborator with Kevin on this project. And this is the FHB Genomic Selection Project at Minnesota. And so for this project, what we have is we have parents from the University of Minnesota breeding program, barley breeding program, and parents from the North Dakota State University barley breeding program. And these individuals were, some individuals from these programs were selected, and they were crossed, okay, to one another. So we had Minnesota by Minnesota crosses, Minnesota by North Dakota crosses, and North Dakota by North Dakota crosses. Okay? And then Kevin derived progeny from these crosses. So we have 100 progeny from each of these different types of crosses for a total of 300 progeny. Okay? And what I really like about this data set for answering questions like this is a data set of a real breeding program. Okay? This isn't some sort of diversity panel that's used for association mapping that somebody just sort of manipulated some way to look at genomic prediction. This is a real breeding program data set. And the individuals we're using here for the validation data set are actually one generation removed from a training population, okay? Which is what we really like for using genomic prediction in an actual breeding program. So this is the relation. This is a heat map of the A matrix or the G matrix, okay? The realized genomic relationship matrix between the individuals in the validation population and the training population. As you would guess, the individuals from the Minnesota training population are more closely related to the Minnesota by Minnesota progenies. Same for North Dakota. And then there's a big mix here in terms of the relationships between the inter-population crosses and the two training populations. Okay? So the point is there's a range in relationships here, okay, in this set. So what we want to look at is we want to look at the relationship between the training population and the selection candidates and how that affects prediction accuracy, or the relationship between that and prediction accuracy. So what I do here is I take a sliding window approach. I take the training population and I sort the whole thing according to its average relationship to the validation population, okay? And then I go through and I take a sliding window approach and I take the first 200 most closely related individuals and I build a model and I go to the next increment by 10 to the next set of 200 lines and build a prediction model and go all the way down to the least closely related individuals to my validation population. And so I get this figure here that clearly shows that there's a relationship, okay, between predictive ability and the mean relationship between the relationship between the average of the training set and the validation set. Okay, so as the relationship increases, my prediction accuracy increases. As it decreases, I have very, very poor prediction accuracy. Okay, and this has been shown before in other studies. So my next question was, as you go further and further out in terms of relationships, does adding increasingly unrelated individuals to your training population ever actually reduce your prediction accuracy? Okay? Can it ever actually hurt you? Even though you're adding more individuals, does your prediction accuracy go down because the individuals are more unrelated? And by taking a similar approach, by sorting the individuals according to their relationships and adding, building my training population size up, okay, by adding the 10 most closely related individuals and the 10 next most closely related individuals and so forth, I've observed for these three traits that, yes, in some cases, you actually do start to head in the opposite direction in terms of prediction accuracy. Okay, so as we start adding, for example, in this situation, we're trying to predict the Minnesota by Minnesota progeny, and as I start adding individuals from the North Dakota program in there, I start to go backwards in terms of prediction accuracy. And there are lots of reasons for this. One could be an allelic effect, some epistasis, so an allelic effect by genetic background interaction. Another reason could be the lack of a common LD phase between the training population and the validation population. Okay? As populations become more diverged, it's more likely that the markers and the QTL are going to be opposite linkage phases. Okay? So I'll have maybe a positive marker effect in this population, but in this population actually should be a negative marker effect because it's linked to the opposite QTL allele. So there's a statistic out there called the correlation of R, which takes this into account. So this takes into account both the marker density and the relationship. And this paper here showed that we have several different types of Holstein populations, or cattle populations of different types of breeds. And you can see that as the marker distance between the adjacent markers increases, the statistic called the correlation of R decreases, and it decreases more rapidly for populations that are more diverged than populations that are more closely related to one another. Okay? So taking this, with this idea in mind, I can look at this relationship between the correlation of R and the mean relationship between the training population and the validation population in this barley situation. Taking a sliding window approach again, and I see something similar. And if I calculate something called the correlation of R, between the whole validation population and every individual in my training population, I can rank them again, sort of like I rank them based on their relatedness, and show that at some point here, I either plateau in terms of prediction accuracy, or my prediction accuracy is actually trending downward. As I add individuals, I have a lower and lower correlation of R with my validation population. Okay? So I like this approach because this statistic takes into account both the marker density and the relationships. Okay? You're still going to have a pretty high correlation of R between distant populations if you have high marker density. But if you have low marker density, it's going to decrease more rapidly. All right, so to summarize all that, genotyping by sequencing works quite well for genomic prediction. Missing data, don't worry about filtering markers from what I can tell. Relationships matter, and again, prediction accuracy can be quite high despite knowing nothing about the genetic basis underlying traits. So it's a very popular approach for breeding. Okay, so let's go back to the topic of this symposium. So I want to break phenotyping down. For my purposes, I want to break phenotyping down into two categories. So we have what I call precision phenotyping and phenomics. Okay? So precision phenotyping is what I think of as using all technologies available to us to maximize the precision of our phenotypes. Okay? Whether that be precision planters or using good descriptions of our field and our landscapes for doing a spatial analysis, or maybe putting our plots on more uniform fields. Okay. So I can see the practical purpose of this. The end outcome of this for plant breeding and for genomic selection is increased heritability. Okay? And we all know that increasing heritability increases our response to selection. Increase in heritability also increases our heritability of our calibration data set, also increases our genomic prediction accuracy. Okay? So I can clearly see the rationale behind that. There's also phenomics, and as Jonathan Lynch pointed out, the phenome is a set of all phenotypes expressed by an organism, or phenes, or however you want to slice it. And if G by E is important, which of course it is, to get a full phenome, we should measure all possible phenes or traits in all possible environments. And I would argue that for dynamic traits that change throughout time, we should probably measure all phenes or traits, at every single time point throughout plant development. Okay. So I have a harder time. The plant breeder inside of me has a harder time trying to figure out how I use this in a breeding program. Okay. For one thing, this is going to take a tremendous amount of cost and time to measure all these traits. Okay. And for another thing, at the end of the day, we need to come up with some sort of way to weight all these traits so we can make selection on something like grain yield under some sort of environmental condition. Okay? And it's not at all clear how we're going to weight all these different traits that go into the whole phenome. Okay? And let me remind you that we've made really good progress in a lot of situations in terms of grain yield by measuring grain yield really, really well and not knowing much about the phenome, and not knowing anything about the genome for that matter. Okay. So the plant breeder inside of me has a hard time with trying to figure out how to deal with phenomes for practical purposes of genetic gain. But let me sort of make an analogy for you and describe why I'm interested in phenomics anyway. I don't have a lot of time to read these days because I have little kids, and between my little kids and my job, I don't have much time for reading. But I do every single month. I make sure that I get through the Atlantic Monthly, and an article here one day caught my attention in this magazine. And this article describes this person named Douglas Hofstadter, who wrote this book called Gödel, Escher, and Bach back in 1983. And so the subject of this book is basically trying to use computers to help us understand how we think. Okay? Writing programs to help us understand how we think. So how do we, what happens inside of our brain when we recognize a face, or what happens inside of our brain when we recognize a letter? Okay? And if we can write programs to simulate this, we'd better understand how we think, and therefore we could make machines think. Okay? So since this time, artificial intelligence has made great leap forwards. Okay? But it hasn't. That great leap forward has not been accomplished through any better understanding of how we think. It's been accomplished through brute force machine learning. Okay? We've simply built really good black box models and fed high-power computers millions of observations and finely tuned those computers until they make associations between complex objects and what's stored inside the computer. Okay? So this is what's been done, for example, in your iPhone. When you tell Siri something, Siri doesn't really understand what you're saying. Siri is simply just making an association between the inflections in your voice and the command stored in its memory. Okay. So I thought the article was very interesting because it really laid out this divergence of artificial intelligence and machine learning. And Douglas Hofstadter is, from what I understand, still working on the fundamentals of artificial intelligence back at Indiana University. And one thing that caught my attention in this article was this quote here by these two authors in artificial intelligence. Okay? So they point out that maybe artificial intelligence has perhaps become too much like the man trying to get to the moon by climbing a tree. Okay, one can make progress all the way to the top of the tree. Okay, so this sort of struck me as I wonder if this is what we're doing in genomic prediction. If we spend all of our time thinking about simply doing genomic prediction on yield, are we just simply going to be climbing the tree in an effort to get to the moon? So this might be obvious to many in the room, but from my standpoint as a plant breeder, it isn't always obvious. Okay, so that brings me around to why should I be looking into phenomics for drought tolerance? And so one of those, of course, is based on my discussion, is the prospect for perfect knowledge. Okay? If we knew the true effect of every single allele in every single environment, we could make perfect predictions across very diverse genetic backgrounds. Okay? And it might get us to the moon, which is where we want to go. The other is that the patterns of water stress greatly vary from year to year. And I didn't do a very good job I'm not going to do a very good job at elaborating on this in my presentation, so I'm very thankful for Maria's presentation before me that did that. Okay? When water stress occurs, it can vary, and it'd be useful to know how every single genotype in the field responds to every single episode of water stress. Okay? Because we're not going to maybe see that exact same pattern the following year, and doing controlled environments for every possible situation is not feasible. All right, so back to drought tolerance. Okay, so drought tolerance obviously is a very complex trait. One of the things that makes drought tolerance so complex is you don't really know what important secondary traits there are for drought tolerance. So for example, is this genotype susceptible and this genotype tolerant? Or is it the other way around? Okay? Perhaps in an extreme, a year where we have terminal drought, this plant here is conserving soil moisture for use later on. But if we have a rainfall that alleviates that drought, then this line over here that's chugging along at full throttle on photosynthesis is going to do better. Okay? So I really like this article that was published a few years ago by Francois Tordeau at INDA, and it says that any trait or any trait-related allele can confer drought tolerance. Just design the right drought tolerance scenario. Okay? So this is what makes drought tolerance so difficult. So, we have numerous secondary traits that have either positive or negative effects. For example, avoiding photosynthesis through somatal closure would maintain soil water. The drawback is that it's going to decrease photosynthesis. Okay? Roots, on the other hand, increased root growth, great for picking up moisture deep in the soils, but it's a competition for carbon. And then the scenarios that you can draw from these is that you have a trait, for example, for increased root growth. You have a positive effect if you do have deep water available, but if you have shallow soils, it's going to be a negative scenario. Okay? So you have all these possible situations. And the way to get around this is to measure, measure, measure. Okay? You want to measure as many traits as you can, as frequently as you can, and have a good handle on the environmental conditions from year to year. All right, and so to do all this measuring, we need, on the number of genotypes we want to measure for a breeding program, you're going to need something high throughput. What we really want to take the heart rate of the plant throughout the entire season. Okay? Of course, plants don't have hearts, but there are a number of things that we can use to tell us what kind of stress those plants are under. Okay? Things like growth rate. Okay? When plants encounter water stress, the leaves stop expanding, okay? Almost immediately. Somatic conductance, okay, is influenced by drought stress. This results in increased leaf temperature. Okay? Leaf water concentration or content goes down, and chlorophyll concentration is affected. Okay? There are other things that, of course, are affected, but these are things that are relatively easy to measure. And the other thing about these traits is that we can use sensors to measure them. Okay? So we can, for example, take reflectance measures in the visible part of the electromagnetic spectrum, and it'll tell us something about the chlorophyll concentration. We can take reflectance data in the near-infrared part of the spectrum, and it'll tell us something about leaf water content. Okay? This is important because if we're going to do this on a regular basis, the measurements need to be non-destructive, and they need to be high throughput. And another nice thing about using sensors to measure these types of traits is that sensors can tell us things that we don't pick up with our naked human eye. For one thing, we can look outside of the range of the visible part of the spectrum. Okay? And if we focus on very specific wavelengths, we can get a better idea of what exactly is going on, classify tissue types by stressed and non-stressed, or diseased and non-diseased. So there are greenhouse platforms out there to do these types of things, notably, most by Lemnitech in Germany that constructs a lot of these things. Okay? So those have some desirable capabilities, but obviously plants grow in the field, and so we want some field-based platforms for making these measurements. And there are some handheld measurements, and as was talked about, there are some tractor-driven measurements. Okay? And so that, of course, the downside of a handheld measurement is going to be more laborious. Okay? And in my opinion, there are pros and cons of all these things. The downside of a tractor-driven platform is going to be increased compaction, and you invade the plots, always running the risk of damaging the plots. Okay? All right, so that was the background. I want to talk about a couple of studies that were conducted in Nebraska over the last couple of years. Like I said, we're just getting our feet wet in this area. So just last year in 2013, we ran a very small study where we had just 5 hybrids. 4 of those hybrids were commercial checks. One was a historically important hybrid called Nebraska 501D. And in this study, we had 4 water treatments, well-watered, and then 2 intermediate irrigation levels, and a dryland treatment. Now, 2013 wasn't exactly a very droughty year. It was pretty cool, so we didn't see a lot of good moisture stress. This is a layout of that study. So we have the different irrigation treatments here indicated by the different colors, with the different hybrids randomized within these irrigation treatments. And like I said, we didn't have huge yield reductions, but we did have a yield reduction. So the well-watered yielded quite a bit more than the dry land, but the two intermediate irrigation levels here weren't that different. Okay. All right, so the first platform I want to talk about is what I'm referring to as the backpack spectrometer system. What this consists of is, I don't know all the details on this. I have to work with other people to do any of this type of stuff. But it's basically a hyperspectrometer system, okay, that's contained in the backpack. And you carry this thing, and you have a wand here that you hover over the crop canopy. And it's a hyperspec system, so this means that we're going to be measuring reflectance at every single wavelength. In this case, actually, the resolution is 0.4 nanometers. So every 0.4 nanometers, we're getting a reflectance data point. Okay? And we're going from 350 nanometers to just over 1,000 nanometers. And the really nice thing about this system is that it includes, so we're measuring what they call upwelling and downwelling radiation simultaneously. And this allows for the adjustment for light conditions to change. Okay? So light conditions are really going to affect the radiation coming off of the canopy, and this makes that adjustment instantaneously. Okay, so once again, inside the spectrum here, we're taking measurements every 0.4 nanometers. And I want to point out that this cannot be done without the help of these folks from CALMIT, which is a center at the University of Nebraska that focuses on remote sensing. Okay? So big thanks goes out to them. All right, so this is what it looks like in terms of reflectance across the entire part of the spectrum. Okay, you can see that for average across all the dry land plots versus the well-watered plots, you can see that we're getting more reflectance in the visible part of the spectrum, and less reflectance up here in the near-infrared part of the spectrum. Okay. Now, one thing we could do is, for these hybrids in this experiment, we have different hybrid levels and different irrigation levels. And you can do an analysis of variance at every single point on that spectrum where this backpack system collected data. Okay? And if I do that, you can see that the hybrid effect is the p-value here in the y-axis. The hybrid effect is generally not significant. Okay? So there's not a single point in the spectrum, except maybe down here around 400, where we're able to pick up differences between the hybrids. Okay? We are, on the other hand, able to pick up differences between the irrigation levels. Okay? So especially in the visible part of the spectrum. Okay? We're telling those water treatments apart. But there are better ways to go about this. And so you can calculate these vegetative indices that have been developed by the folks working in remote sensing and plant physiologists. And since we're taking reflectance data at every single point along the spectrum, we can pretty much calculate any vegetative index that you can imagine. These are a few that we chose. We have a normalized difference vegetation index, a very common one to use, NDVI, gives a general picture of the canopy coverage. Normalized difference red edge relates to chlorophyll concentration. Canopy chlorophyll content index is the ratio of those two things, and so it gives a better handle on chlorophyll per unit of canopy. And there's this PRI, photochemical reflectance index, which is a little more detailed and actually relates to xanthophyll activity, and the Gittleson red edge index. Okay? So these are those indices calculated for the different water treatments and for each of the hybrids. And you can see that there are differences between the water treatments with the direction across the water treatments going in the expected direction. But the ANOVA here down here tells us that, again, we're not picking up any differences among the hybrids for these indices, but we are seeing big differences between the irrigation levels. And this is for July 26th. Okay. For August 19th, again, for two of the indices we looked at in more detail, we're not seeing differences between the hybrids. For the irrigation levels, we are seeing big differences. Okay. So let me tell you something about a UAV that we're working with. And this is an OctoCopter platform that was developed at the University of Florida. and it can carry up to about 1.5 kilograms. And to the bottom of this UAV, a tetracam is mounted. Okay, a tetracam that has 6 bands, one at 530, 670, 710, 760, 800, and 970. Okay, and these are chosen because they correspond to specific regions of the spectrum that relate to canopy water content and nitrogen status. So if we fly it, so during both the summers of 2012 and 2013, Richard Ferguson, who's a soil scientist at UNL, his group basically flew this thing. They have a lot of expertise in using UAVs for evaluating fields. Okay, so I needed to work with Richard Ferguson to pull this off. So he flew this above my fields during 2013. This is exactly the same study that I just talked about on the backpack system. Okay, so this is what it looks like from above, and this is what the image looks like in those wavelengths. So what we did is, we had to take two images to capture the entire field at about 70 feet above the canopy. Okay? And so what Chelsea Wu, who is a specialist in GIS, what she was able to do is she was able to take these 2 images and stitch them together. Okay? And then she's able to make what's called a shapefile. And the shapefile basically consists of little boundaries for each of our 4-row plots. And from each of these 4-row plots, then, she extracted out all the reflectance data, okay, contained in those pixels. Okay, and then she can give it to me in an Excel sheet and something I can actually work with. So we get the reflectance values from those images, and we calculate, this is the crop canopy, chlorophyll crop canopy index, the one that was calculated as before with the backpack system. So for this, what we have here is we have the green is dry land, and the purple is well watered. And so you can see here now, in terms of the differences between the different hybrids, we're actually seeing some differences. Okay? And we're also seeing differences between the water treatments. Okay? If we average across the hybrids for this index. All right, so to summarize what we found in 2013, okay, we saw differences between the water treatments with the backpack hyperspec system. We didn't see any genetic variation or any variation among the hybrids. However, when we used the multispectral sensor on the UAV, we were able to see differences between the hybrids and the water treatments. Okay? And there are numerous reasons for this. One might be that it takes quite a long time to go from the first part of the field to the last part of the field, and conditions have changed throughout that whole time. So you have some variation throughout time with the backpack system. On the other hand, UAV takes an image of the entire field simultaneously. Okay, so that's a very desirable property of that. All right, so that's Lincoln 2013. Like I said, not a very good drought year for doing drought studies. So let's go to western Nebraska, and let's go back in time in 2012, where we actually had a really good drought. Okay? So we're going to go to Brule, Nebraska, which is close to the Colorado border. And I should point out, though, if you haven't heard this line, I should make sure you know it, that the water gradient from western Nebraska to eastern Nebraska is very, very steep. We go all the way from about 12 inches out here to about 33 inches per year in the eastern part. Okay? So we have great opportunity in Nebraska to look at variability in terms of rainfall in the genotype by environment interaction. All right, so in Brule, Nebraska in 2012, we planted out 98 top crosses. Okay? These were inbred lines that were top crossed to 2 commercial testers. This wasn't from a diversity panel or anything like this. This is from a set of inbred lines that are historically important inbred lines, expired PDP inbred lines, things like that. So things that are well adapted. We had 2 water treatments. We had a full irrigation level and then 40% of that full irrigation level. Okay? And in 2012, we saw really good differences between our 2 water treatments. We had a 40% yield reduction, okay, in the water-stressed environment. So this is what we want to see. And it was so droughty out there that if those plots weren't irrigated at all, they would have simply died. This is what the study looks like in an image from a manned aerial vehicle. So we have the well-watered, water-stressed, well-watered, water-stressed, separated by about 30 or 40 feet. to allow the variable rate irrigation system to switch over its irrigation levels. Okay. And it was a really big study. There's lots of replication within each of these watering blocks. Okay. And this study was not designed for aerial imaging either. All right, so soil water content here, measured by watermark water sensors. You can see good differences between the water stressed and well-watered treatments. Okay? And we saw some differences out there in terms of genotype by water or irrigation level interactions. We had some genotypes that were rather unstable under water stress conditions. Okay? And we had some genotypes that were relatively stable. Okay? These genotypes yielded about the same under well-water conditions, but these genotypes here on your left yielded about close to only half of what the more tolerant genotypes yielded. Okay? So out there during 2012, Richard Ferguson and his student brought out the OctoCopter, and they imaged this study. Okay? So this is what I mean by this thing wasn't designed for aerial imaging. So it started all the way up here. So I should point out, when this OctoCopter, when it's in the air, it is truly a high-throughput phenotyping machine. It's unbelievable. But getting it up in the air and to take all these images was challenging. And in the battery life, too. I mean, we went through about 6 batteries to go around this whole thing. So that's a huge limitation of UAVs is batteries. I mean, until they make a quantum leap forward in battery technology, we're not going to be able to use these things for very long periods of time and do constant monitoring of our field plots like we would like to. Okay, anyway, so we took all these images across the entire study, and Chelsea Wu, very talented person in GIS, was able to actually stitch all these together, which I thought was an amazing feat. So they're all stitched together, and then she extracts out the plot data and gives the data to me in an Excel spreadsheet, and we do a little bit of analysis, and we calculate some of the indices that we calculated before, NDVI, this chlorophyll green index, this chlorophyll index here again. And we see big differences between the water treatments. These differences are all statistically significant. And we see quite a lot of variation here among the hybrids in these watering blocks. Okay? And a lot of people have asked about repeatability and heritability and those types of things today. I actually calculated the heritability for this set of 98 top crosses. And the heritability isn't too bad for these indices. Okay? So, and it's a little bit better under water stress conditions, I presume, because we're seeing more variation for these canopy properties when there's a little bit of water stress compared to well-water conditions. And maybe this isn't why we're seeing big differences in 2013 either, because it was a relatively wet year. All right, so summary from this study is that we successfully flew the OctoCopter. We're detecting variation, so we're able to work together, extract out data, detect genetic variation for these types of traits, okay, for these canopy indices. And I want to point out that this was accomplished by a bunch of soil scientists and plant breeders and minor detail, a GIS specialist. But and Richard Ferguson does have good experience with UAVs, but at the end of the day, we're still trained as agronomists and plant breeders. Okay. So if we really, really want to get fancy with this type of stuff, we need to start working with other people. And we're fortunate at UNL to have what we call, or what they call, the Nimbus Lab. In other words, the UAV lab at UNL, led by Carrick Detweiler and Sebastian Elbaum. Okay? And they specialize in making UAVs do very complex tasks. Okay? So to get these guys to work with you, you can't simply say that you want to go take some pictures of some field. This is not challenging enough for them. What they would really like, if I were to say, I'd really like UAVs to hang tassel bags. Then they would get right out there and they would be fired up by the job. Okay? But one thing, the payoff in terms of amount of work that would take to the amount of work it would save isn't that great. And I happen to like hanging tassel bags anyway. So I'm starting some collaborations with these guys. And one of the things that we're looking at doing is using these micro UAVs for measuring plant height. Okay. So this is the idea. It was a very early phase, although the algorithm for doing this is soon to be published in the conference proceedings. But the idea is that take a laser scanner mounted to the UAV, scan the crop canopy, Every once in a while, the laser scanner hits the top of the plant, so you can infer that that's going to be at the top of the plant. Okay? And every once in a while, the laser scanner hits the ground. Okay? So you can infer that to be the ground. And so you can take the difference between the two, and that'd be the plant height. Okay? So this is the algorithms have been developed by Dave Anthony, who's a graduate student in computer science. And I can't take credit for any of this kind of stuff. I literally just took them to the cornfield and showed them some corn plants. But we're going to design some studies for next year where we're going to look at some specific differences between different methods and looking at relative differences in genotypes in a planned study. All right, so I don't have, we haven't been working on this very long, so I really don't have any conclusions. I have perspective, is I guess one way to put it. Okay, so we want to make routine measurements for understanding how all the genotypes respond to every single environmental condition encountered throughout the entire year, to have that fuller understanding of stress tolerance. Okay? And to do this, we're going to need highly automated, non-destructive measurements. Okay? So moving in these directions, working with engineers to get these technologies off the ground is definitely needed. Okay? And we made some progress using UAVs for this purpose. There's a tremendous amount to do, and we're going to be needing to work with these engineers to do even more. Okay? I think, I'm not sure if Pat Schnabel's in the room or not, but last December I heard Pat Schnabel say at the ASTA meeting that he loves working with some of these engineers because you give them a really, really big problem and they set down to solve it no matter what. Okay? You give a biologist a really, really big problem and they say, isn't that a very fascinating problem to have? And I would go a step further and say that these computer scientists are they're like wizards. I mean, the things that they can do with technology are just amazing to me. And so we really need to get them on board for these problems. Another perspective I want to add is that I really think that the technology is way ahead of the questions, and the technology is way ahead of the physiology. Okay? And so we've been sort of chasing the technology, and next summer we're hoping to go back and do a little better job in the physiology and relating these vegetative indices and things we can measure with these sensors to real ground truth measurements that mean something to plants. So I have two sets of acknowledgements. I have my genomic prediction acknowledgements. I have a grant for Natalia in maize silage. The wheat work has been done by Steve Menziger, Ibrahim, Jesse Poland, Edward, and some graduate students, Mary and Catherine at UNL. Kevin Smith, of course, is a great collaborator at Minnesota. Soybean, George Graff and Diego and some of his students. And the drought tolerance work, I sort of have grouped here by different areas. John Leachins and Leacott are students of mine, or Leacott's a postdoc. OctoCopter work has been headed up by Richard Ferguson, who's a great collaborator at UNL, and his student Brian Krenke, and of course Chelsea. The Nimbus lab I mentioned, and helping me get these field trials, especially in western Nebraska, was accomplished by Greg Kruger, who was stationed in North Platte, Nebraska, and Tim Shaver. And the hyper-spec work was helped by Catherine, Mary, Steve, and some folks from CALMIT. Okay? And with that, I'd like to thank you and take any questions.
Questions for Dr. Lorenz. Please use either the microphone in the center of the room or the one I'm holding here so that the people in cyberspace can hear us.
I think you may have already said this, but was the spectral range of the OctoCopter the same as the backpack?
No, the OctoCopter just had 6 bands, 510, 630, one in the 700s, and a couple in the 900s. The backpack went all the way from 350 to about 1,000 every 0.4 nanometers. So the range is a little bit wider, but the resolution is a lot higher.
It's notable that with less spectral information, the OctoCopter still beat the backpack, I guess.
Yeah. I'm a fair bit surprised, but we'll repeat this stuff next year and see if we come up with the same result.
I was interested in the raw spectral data where you weren't computing vegetation indices. And as you mentioned, the computer scientists might have a lot to offer. And is there going to be any investigation into the raw spectral data without computing vegetation indices, without any a priori knowledge of what those particular wavelengths reveal about plants. Maybe we'll discover some new indices or new ways to find things out about the crops under stress.
Yeah, absolutely. I mean, one thing to do would be to take sort of a near-infrared approach like we do in the lab, and just, measure something we're interested in the field, and then use a hyperspec to get all the reflectance for all the different wavelengths, and then do just a calibration. That's one approach. So there are lots of opportunities there.
It seems to me one of the themes that we've seen so far is that it's hard to choose the right index.
Yeah, that's right. That's right. And it's really, I mean, not being educated in this area, it's really bewildering to get into this literature, and so there's a lot out there to learn.
So my question is more on the logistics side or experimental design side. So when you're screening for when you're screening for drought tolerance in the experimental setup like the pivot irrigation that you had, when you say 40% water, what was the actual irrigation level based on? Was it like a bulk for the whole field, or actually was it separated down by each of the different lines you were trying to screen? Did they have the same amount of irrigation for each one? How was that set up?
There's I don't know what the actual irrigation level was. I mean, it depends on how much precip there is during the year, but there was a standard irrigation level set that they were using for that field for production purposes. Mm-hmm. And then the 40%, those water stress blocks were just 40% of whatever that irrigation level was.
Do you think, when I think about setting up experiments in this way, do you think it might actually penalize the more conservative crops that may be actually using less water, maybe able to be a little more drought tolerant when you're giving them all the same amount of water? So you're not basing the actual stress on the soil moisture content. You're basing the stress on the amount of water being put in. So if the amount of water going out of each genotype is not the same, then they're not actually experiencing what they would be experiencing if they were, say, grown in a bulk field. So the more conservative water users might actually be penalized in a system like that. I guess, do you think that's a possibility?
Yeah, that's right. And this is a very specific water stress scenario. Okay, and then this is a scenario does not resemble dryland farming very well. Okay, but Nebraska farmers are interested in bringing down their irrigation levels. If they could irrigate at half the rate that they do, they would like to do that. And that's a specific water scenario, drought scenario. So, yeah, I agree, though. It's not very well defined.
Are there any other questions from the audience? And I have one here from cyberspace. They ask, how well has the microcopter plant height algorithms worked so far, and what kind of camera did that use?
It's a laser scanner, some sort of LiDAR sensor. And the precision right now, they can measure absolute plant heights of a single plant within 3 inches. From what we went out and measured some plants, and they did their scanning and developed an algorithm, estimated plant height. So, yeah.
Any more questions for Dr. Lorenz?
I should point out we haven't looked at relative differences or heritability or repeatability or anything like that among multiple genotypes yet.
Well, let's thank Dr. Lorenz for coming to speak.